Hi all, welcome to a new session. So in this video module, we are going to discuss about the histology. Okay, so we'll see what are the topics which we need to see. First, we will be discussing about the epithelial tissues. Okay, then we'll be moved to the glands, connective tissue, the cartilage, the lymphoid, the integumentary system where you will see the skin, the cell junctions, the muscular system, tissue, respiratory system, digestive system, urinary system, and the genital system. So first we will see the common histology and then we will move on to the respective system. Okay. So moving on to the epithelial tissue. Based on the number of layers, okay, the epithelial tissue is divided into simple epithelium and compound epithelium. Okay. From the name itself, it is clear. Simple epithelium means it has only a single layer. Okay, single layer. Compound epithelium, it is multi-layered. Okay, multi-layered. Okay. So the first simple epithelium is the simple squamous epithelium. Okay, simple squamous epithelium. Then you all know what are the features of a simple squamous epithelium. We know that it is the cell. If you take this cell, it has more breadth than the length. Okay, so the breadth will be more than the length, right? And what about their nucleus? They will have flat nucleus. Okay, the function of this simple epithelium is exchange of substance. Okay, it can be gases, it can be fluids. Okay, so the function of the simple epithelium is mainly exchange. That is why we can see simple epithelium where it is seen in the bronchi, not bronchi, sorry, it is seen in the alveoli. Okay, the alveoli, then the blood vessels. Okay, then the body cavities, right, and the Bowman's capsule. Okay, so these are the places where you can see simple epithelium. Okay, right. So this is about the simple epithelium. Now coming to the simple cuboidal epithelium. Okay, that was simple squamous epithelium. Now simple cuboidal epithelium. Simple cuboidal means what? The breadth of the cell will be equal to the length. Okay, so you will have a uniform cell where the breadth will be equal to the length. Okay, and they will have round nucleus. Okay, round nucleus. Can you see here? This is a simple cuboidal epithelium, right? The shape is almost square shape and they have a round nucleus. Okay, now what's the function? The function is secretion. Okay, simple cuboidal epithelium is for secretion. Okay, now tell me where you can see this simple cuboidal epithelium. The location is the thyroid follicle. You can see in the thyroid follicle. Then the germinal epithelium of ovary, and the kidney tubules. Okay, kidney tubules. Actually the DCT. Okay, so this is about the simple cuboidal epithelium. Moving on to the simple cuboidal epithelium with microvilli. Where can you see a simple cuboidal epithelium with microvilli? Answer is you can see the kidney tubules. Which tubules? In the PCT as well as in the thick descending limb. Okay, thick descending limb of loop of Henle. Okay, where you can see this is Henle's loop. Okay, or loop of Henle. Okay, loop of Henle. Okay, so here you can see simple cuboidal epithelium with microvilli. Okay, moving on to simple columnar epithelium. Simple columnar epithelium. Here, what about the features? Here, the length of this cell will be more than the breadth. Okay, length will be more than the bread, okay, and they have an elongated or an oval nucleus, okay, elongated nucleus, and the function is secretion itself, okay, secretion. Now what can you see? It is same location. Location is the stomach and large intestine, okay, stomach and large intestine. Okay, so this is about the simple columnar epithelium. Now moving on to simple columnar epithelium with what? With microvilli. Where you can see simple columnar epithelium with microvilli. Answer is in the small intestine. Okay, in the small intestine you have simple columnar epithelium with microvilli. This microvilli helps in absorption, right? Then next is ciliated columnar epithelium. We know that this cilia will helps in what? It will helps in movement, okay, mobility, right? So the cilia will helps in movement. So where you can say the ciliated columnar epithelium, the location is at the fallopian tube and the eustachian tube, okay, and the eustachian tube, okay, where you can see ciliated columnar epithelium, right? Now moving on to the pseudo stratified columnar epithelium. What do you mean by pseudo stratified columnar epithelium? Here, due to the difference in the level of the nucleus, okay, one nucleus is here, one nucleus is here, next nucleus will be here, right? So the difference in the level of the nucleus, we may suspect that it is a multicellular or multi-layered epithelium, but actually it is a single layered epithelium. Why? Because all the cells, okay, all the cells will be attached to the basement membrane. All cells attached to the basement membrane, okay, basement membrane, right? And where you can see this pseudo stratified columnar epithelium? Answer is you can see at the this is the respiratory epithelium, which is present in the trachea, the bronchus, okay. So these are the place where you can see pseudo stratified, okay, and usually the pseudo stratified, they will have what? Cilia also. So pseudo stratified ciliated columnar epithelium you can see in the respiratory tree, that is trachea and bronchus. I'll show you the figure. Can you see here? The nucleus are in the different level, but all the cells are attached to the basement membrane. Okay, this is the pseudo stratified ciliated columnar epithelium which you see in the trachea and bronchus. Okay. So this is about the simple epithelium. Now we are moving on to the compound epithelium. Compound epithelium, I told you there will be many layers, okay, many layers of cells. Okay, so based upon the apical cells, okay, what is the cells that comes at the apex? We have divided it into not simple, it is compound, or we will call it as the compound epithelium, we will call it as stratified epithelium, okay, stratified epithelium or compound epithelium. So if this cell in the apical region is a squamous cell, we will call it as stratified squamous epithelium. Understand? That is, if the apical cell is a squamous cell and we have many layers of cells, we will call it as stratified squamous epithelium, right? In stratified squamous epithelium, the cells that is present outside, usually the skin, okay, they will have an external deposition of keratin, okay, the skin or the cells that is present in the external surface, okay, they will have an keratin deposition. This is to protect the cells, okay. So if there is keratin deposition over the squamous cells, we will call it as the keratinized stratified squamous epithelium. Understand? We'll call it as keratinized stratified squamous epithelium, right? If the keratin deposition is there, we will call it as keratinized stratified squamous epithelium. Where you can see keratinized stratified squamous epithelium, it is present in the skin, okay, in the anal canal, okay, in the distal part of the anal canal, right? Now, moving on to the non-keratinized stratified squamous epithelium, right? So if there is no keratin deposition in case of the stratified epithelium, we'll call it as non-keratinized stratified squamous epithelium, or simply stratified squamous epithelium. So you can see this type where there is actually the squamous cells, okay, squamous cells in the apical region, and under that you have multi-layered cells, okay, but there is no keratin deposition. You can see such type of cells in the oral cavity, right, in the tongue, the tonsils, okay, then the vocal cord, the epiglottis, okay, the oral surface of the epiglottis. So these are the area, and in the vagina. Okay, so these are the area where you can see stratified squamous epithelium. And what is the function of this stratified squamous epithelium? This is act as a barrier or protection. So many layers are there, right? So in case of vocal cords, even though it is an internal laryngeal structure, why? Because the vocal cord is a structure which will move frequently, right? So there are chances the wear and tear can occur to vocal cord. To prevent such wear and tear, vocal cord is provided with stratified squamous epithelium. Understand? So this is about the stratified squamous epithelium. Now we are moving on to the stratified cuboidal epithelium. Okay, cuboidal epithelium. That means the apical layer is what? Apical layer is cuboidal cells. Okay, apical layer is cuboidal cells. Okay, and where you can see this stratified cuboidal epithelium? In the exocrine glands. Okay, the exocrine glands have this stratified cuboidal epithelium, right? Moving on to stratified columnar epithelium. Stratified columnar epithelium. Okay, so the apical layer will be columnar cells. Okay, where you can see it is present in the membranous urethra. Okay, membranous urethra in male. Okay, and the penile urethra. Okay, so this is the area where you can see stratified columnar epithelium. Moving on to the very important one, that is transitional epithelium. Transitional epithelium. What's the specialty of this epithelium? Here, the epithelium will transform from one form to another. Okay, there will be transition of epithelium from one form to another. Okay, it is a multi-layered and transition from one form to another, and usually the apical layer has umbrella shaped cells. Okay, umbrella shaped cells. Okay, and due to this transitional epithelium, the cells or that organ, they will get a property of stretchability. Okay, stretchability will be is due to this transitional epithelium, right? So I'll show you the transitional. Can you see here? This is the umbrella cells, umbrella shaped cells. Can you see the umbrella shaped cells? So umbrella shaped cells are present in the apical region. Then you can see the columnar cells. You can see the in between you can see the cuboidal cells. Okay, so there will be one form of epithelium will be transformed to another form of epithelium. Right now, where you can see which is the location. Location will be from tip of the collecting duct. Okay, the distal part. Okay, collecting duct, tip of the collecting duct up to the prostatic urethra. Okay, up to prostatic urethra. All the structures that comes in between. Which are the structures that comes in between? It is the renal calyces. Okay, the major and the minor calyces. Then renal pelvis. Then the ureter, the urinary bladder, and the prostatic urethra. Okay, here you have pyramids. Okay, renal calyces, then you have the pyramids. Okay, so all these structures will form transitional epithelium. Understand? So this is about the transitional epithelium. With this, we have completed the epithelium part. Okay, one thing I need to, I'll like the exceptions. Okay, I'll tell you. There is something called as para-keratinized epithelium. What does mean by this para-keratinized epithelium? That is, even though there is keratin deposition is there, and inside in that keratin deposition, at the apical layer, you can see the nucleus. Okay, you can see the nucleus in the apical layer. Okay, such type of epithelium where there is keratin deposition and the apical layer, you can see nucleus, that is called as para-keratinized epithelium. And can you tell me why you can see this para-keratinized epithelium? It is seen in the hard palate. Okay, hard palate. This is one exception you need to know. The second one is about the squamo-columnar junction. Okay, squamo-columnar junction. What is meant by squamo-columnar junction? This is a transformation stage. Okay, from the columnar cells, the epithelium will transform into squamous. Understand? And there will be a junction where this transition occurs. Okay, what is the specialty of this junction? There are increased chances of metaplasia at this junction. Okay, because it is transitional stage. Okay, so increased chance of metaplasia, which can lead to dysplasias and can lead to cancer. Understand? That is about this increased chance of cancer development. So where you can see this squamo-columnar junction, it is present in the esophagus, right? Then in the uterine cervix and the anal canal. Okay, in these three areas, you can see this squamo-columnar junction. Understand? Right. So this is about the epithelial part. Now we are moving on to the glandular parts. Okay, about the glands. The glands, how these glands are formed? The glands are formed when there is invagination of the epithelial cells into connective tissue. Okay, so this invagination of the epithelial cells into connective tissue resulting in the formation of glands. Okay, there are two parts for this epithelial cells. Okay, one is the secretory part, okay, which forms the gland, and then there is the conducting part which forms the duct. Understand? So if the secretory part of the epithelium will forms the gland and the conducting part of the epithelium will forms the duct. Okay, right? So there are two types. Okay, broadly speaking, there are two types of glands. Okay, one is the exocrine gland. What is meant by exocrine gland? These glands, they will have both glandular part as well as the duct part. Then you have the endocrine glands. What do you mean by endocrine gland? These are the ductless glands. Okay, they don't have any ducts. Okay, the conducting part will disappear after invagination. That is called as the endocrine glands. Okay, this is the first classification. Now I am moving on to the endocrine glands. Sorry, exocrine glands. Sorry, exocrine glands. I told you these are the glands which have duct. Okay, the first classification is, what about if the glands can be unicellular. Okay, that means only a single layer. Okay, that is called as the unicellular glands. An example of unicellular exocrine glands are the goblet cells. Okay, goblet cells. I'll explain about goblet cells later. Then it can be multicellular. Most of the exocrine glands are multicellular. Okay, right. Now the secretions of the exocrine glands, they are called as enzymes. Okay, they are called as enzymes. Understand? Now coming to the second classification. Second classification, if there is only a single duct, okay, a group of cells, and they are releasing the secretions into a single duct, such gland is called as a simple gland. Okay, simple gland means what? Only a group of cells secreting their secretions into a simple duct. Okay, one duct. Then you have compound gland. What do you mean by compound gland? Compound gland means there will be a group of cells which will release its secretion into one duct, another group of cells which will release its secretion to another duct, another group of cells which will release its secretion to another duct, and all these three ducts or all these ducts will join together to form a major duct. Okay, so there will be multiple ducts. Such type of glands are called as compound glands. Okay, so this is about the introduction about exocrine glands. Now, endocrine glands. I told you endocrine glands means what? They have no ducts. Okay, so they'll be releasing their secretions. Okay, secretions are released directly into the bloodstream. Okay, and the secretions are called as what? Secretions are called as hormones. Understand, they're called as hormones. Okay, how they become endocrine? Because I told you there what had happened. The duct part, okay, the conducting part has disappeared after invagination. Okay, and these endocrine glands, they are highly vascular because their secretions are directly going into the bloodstream, so they are highly vascular. Okay, so this is about the exocrine endocrine introduction. Now coming to the first type of exocrine gland, that is the goblet cells. Okay, goblet cells, these are, I told you, these are unicellular exocrine gland. Okay, and what is the secretion of the goblet cells? It is mucus. Okay, goblet cells will mucus, and the method of secretion is exocytosis. Can you see here the exocytosis happening? Goblet, the mucus is secreted out. Okay, and since it is secretion is mucus, if you take the goblet cells, they will stain pale because the mucus doesn't take up any stains. Okay, so they will stain pale with H and E staining. Okay, the eosin hematocylin staining, they will stain pale. Okay, so this is about the goblet cells. Okay, now moving on to the next classification of the exocrine glands. Okay, symbol glands. So depending upon the glandular part, okay, depending upon the glandular part, the symbol glands are divided into symbol alveolar. Alveolar means what? Cuboidal cells. Okay, that is called as alveolar. Okay, symbol alveolar. And where you can see symbol alveolar, actually there is no adult gland, that is symbol alveolar. Okay, next is symbol branched alveolar. Okay, symbol branched alveolar. Okay, that means one here, you have one here, one here. Okay, this forms the alveoli. Okay, and they will all will opens into a single duct, that is symbol branched alveolar. Where you can see symbol branched alveolar, it is seen in the sebaceous glands. Okay, sebaceous. Then symbol tubular. Okay, so here it is in the form of a tube. Gland is in the form of a tube, symbol tubular. The intestinal glands are symbol tubular. Okay, intestinal glands are symbol tubular. Okay, so can you see here, this is the branched alveolar. Okay, symbol branched alveolar. Alveolar, or we call it as acina. Okay, alveolar or acina. Then now the next is symbol coiled tubular. Symbol coiled tubular. Can you see here, symbol coiled tubular? It is like a coil. Okay, the tube forms a coil. This is the merocrine sweat glands. Okay, merocrine sweat glands are symbol coiled tubular glands. Last is the symbol branched tubular. Symbol branched tubular glands. Okay, right? The mucus glands. The example is the mucus glands of the duodenum, the lungs. Okay, they will all form what? They will all form symbol branched tubular glands. Okay, so this is about the simple glands. Okay, now we are moving on to the compound glands. Compound glands, I told you, it is here a group of cells. They will be draining into a duct, and this duct will drain into some larger ducts. Okay, they are called as compound glands, right? So if you take the secretory part, okay, the secretory part, the secretory part can be alveolar or acinar, okay, or it can be tubular. And the secretory part of the compound glands can be alveolar, acinar, or tubular, right? So based on that, the compound glands are divided into compound alveolar, which you can see in the mammary glands. The mammary glands are compound alveolar glands. Then compound tubular glands. Compound tubular means tubular. Okay, the glands will be tubular. These includes the bulbo-urethral glands, then the testis. Okay, so these are the example for compound tubular glands, and it can be compound tubulo-alveolar gland. That means here both the secretory part can be tubular along with alveolar. Okay, so both type of secretory part will be present in case of compound tubulo-alveolar gland, and it is seen in the salivary glands. Okay, so this type of glands are seen in the salivary gland. Understand? I'll show you. Don't worry, I'll show you. Can you see this is the compound alveolar? Okay, so the alveolar part, and the secretory part will forms the alveolar part. Okay, the secretory part is the alveolar part. Then this is the individual duct, and this is the common duct. Okay, so here the secretory part is tubular and alveolar. Can you see here? So it is the compound tubulo-alveolar. Okay, so when it comes here, it is tubular. Okay, so example is this is mammary gland, this is salivary gland, this is the bulbo-urethral gland. Okay, then you have the testis, which part? Seminiferous tubules. Okay, so this is about the compound glands. Okay, compound glands and simple glands. Okay, now based on the secretions, okay, based on the secretions, the nature of the secretions, the glands are divided into serous glands and mucinous gland or mucus gland. Serous glands means what? They will have serous secretion, or they will have thin watery secretion. Okay, the serous secretion. And mucus gland, they will have thick mucoid secretion. Okay, so we will take each one. First about the serous gland. Okay, this is the serous gland. Example is the parotid salivary gland. Can you see the cells? What type of cells? They will have pyramidal cells. Okay, pyramidal cells with round nuclei. Okay, can you see the round nuclei? And can you see the apex of the gland is very bright pink? Okay, that is called as apical eosinophilic. Okay, apical eosinophilic. That means what? The secretions will concentrate at the apex. Okay, okay, that is called as apical eosinophilic due to the collections of secretion at the apex. Okay, and the example I told you, it is the parotid gland. Example is parotid gland. Okay, moving on to the mucus glands. Okay, mucus glands. We know the mucus gland is mucus secretion. Mucus they will stain pale. Okay, mucus glands will stain pale. Okay, and the type of cells, what type of cells you can see here? You can see cuboidal or columnar cells. Okay, columnar cells. Okay, due to this mucus secretion, the nucleus is pulled to the periphery. Okay, nucleus is pulled to the periphery. Okay, the lumen of the gland will be wide. Okay, wide lumen. Okay, right? So this is about the mucus glands, and where you can see the sublingual glands. Okay, sublingual gland as the example of mucus gland. Okay, now coming to the mixed glands. Okay, so here you have a gland where you can see both serous secretion as well as, or serous gland as well as the mucus gland. Okay, both type of glands are present. Okay, and which is the example? Example is this submandibular glands, right? So if you take the mucus gland, okay, can you see here what you can see? The mucus glands, if this is the mucus cells, they will be overlapped by the serous cells. Okay, overlapped by the serous cells. This is called as serous demilunes. Okay, serous demilume. Okay, the mucus cells will be overlapped by the serous cell. And why this occurs? This is to decrease the thickness of secretion. To decrease the thickness of secretion, because we know the mucus secretions are very thick. If what happens, if the mucus secretions are mixed with the thin serous secretion, then the thickness of secretions will be reduced. Okay, and the serous demilunes are seen in which gland? In the submandibular gland. Okay, in this submandibular salivary gland. Okay, so this is about the base classification based on their secretion. Now we are moving on to the classification based on the mode of secretion. Based on mode of secretion. Okay, the first one is the merocrine gland. What do you mean by merocrine gland? Here the cells, okay, even after the secretion, the glandular cells will be intact. Okay, after the secretion of the enzymes or whatever be the glandular product, okay, the cells will be intact. Okay, so the secretions will come out in the, or what's the process of secretion? It is exocytosis. Okay, exocytosis. Okay, now you can see it is, example is the sweat glands and pancreatic glands. Okay, or the pancreas. Okay, okay, so these shows what the cells will be intact. Can you see here? The cells are intact even after the secretions. Okay, first type, that is the merocrine glands. Second one is the apocrine glands. Here the secretions will concentrate at the apex of the cells. Okay, this is the glandular cell. So secretions will concentrate at the apex, and how the secretions are released? By pinching of the apex. Okay, so the apex will form what? They will form blurbs. Understand? Right? So the cells will not be intact. Right? The apex of the cell get pinched off. Okay, pinched off. It's pinched off. Understand? The apex of the cell will get pinched off. Okay, that is how that is a mode of secretion. Okay, the apex get pinched off, or the cells are the secretions are released by pinching of the apex. Understand? Apex is pinched. And what is the example? Example is the atypical sweat glands. Okay, atypical sweat glands which you see in the axilla and pubis. Okay, axilla and pubic area. Okay, that is the apocrine glands. Then mammary glands. Okay, they are apocrine glands. Okay, mammary glands. And you have the ceruminous glands. These are the modified sweat glands. Okay, ceruminous glands. Okay, so there you can see the apocrine glands. Okay, now next is the holocrine glands. Okay, holocrine glands. Actually, this even the gland will have multiple cell layers. Okay, and the cell will be entirely disturbed or damaged by releasing the enzymes. Okay, so the whole cell will disintegrate. Okay, whole cell disintegrate to release the secretion. Okay, and I told you it is a multi-layered. Okay, so the basal cell layers. Okay, basal cell layers, they have this mitotic property. Okay, so they will replace the cells that have disintegrated. Understand? Holocrine glands, and the example is the sebaceous gland. Okay, sebaceous gland. Right? So we have seen so many classifications in the glands. Okay, first we have seen based on the number of cells, unicellular and multicellular. Then we have seen the simple glands, the compound glands. Okay, based on the ducts. Then based on the mode of secretion, we have seen serous gland and mucous gland. This nature of secretion. Then mode of secretion, we have seen the merocrine gland, apocrine gland, and holocrine gland. So this is about the glandular part. Understand? Next, connective tissue. Connective tissue, we know it gives support. Then it connects different tissues. Okay, connects different tissues. Okay, so this connective tissue contains two components. Okay, mainly two components. One is the fibers, that can be collagen or elastin. Then you have the cells. Okay, so these are the components of connective tissue. One is the fibers, and next is the cells. Okay, first we will discuss about the collagen fibers. Okay, collagen fibers, they are long, wavy. Okay, long, wavy fibers which forms large bundles. Okay, forms large bundles, and non-branching. Okay, they doesn't branch, non-branch. Okay, right? And they appear white. Okay, appear white. And in H&E stain, okay, H&E stain, they take up pink color. Okay, pink color. Okay, then is there any special stain for collagen? Yes, the Mason's trichome, they will appear blue. Okay, in Mason's trichome. And von Gissen stain, they will appear pink. Okay, so this is the special states. Okay, now classification of collagen fibers. There are 5 classes. Okay, the type 1 collagen is seen in the tendon, ligaments, aponeurosis, then the dermis and the fibrocartilage. Okay, so these are the sites where you can see type 1 collagen. Type 2 collagen is seen in the hyaline and elastic cartilage. Okay, type 3 collagen is also called as the reticular fibers. Why? Because they are short, wavy, and they forms branches. Okay, they anastomosis with each other. Okay, forming a reticle. This type of fibers are seen in the lymphoid organs. Okay, in the lymphoid organs, that is the lymph node, spleen, liver. Okay, so the lymphoid organs except the thymus. Okay, so except the thymus, all other lymphoid organ have reticular fibers. Okay, then the type 4 collagen, that is seen in the basement membrane and in the lens capsule. Okay, lens capsule, right? So I'll discuss a clinical condition here. One clinical condition, that is Alport syndrome. What is Alport syndrome? It is due to the defect in type 4 collagen. Okay, type 4 collagen, Alport syndrome. Okay, so this is about the classification of collagen. Now moving on to the second fibers, that is the elastin fibers. These are short, thin, non-wavy fibers. Okay, and they form branches and anastomosis. Okay, elastin fibers. Okay, they form branches and anastomosis. Okay, so can you tell me an example where you can see a thick elastin fiber? Thick elastin fibers are seen in the ligamentum flap. FLAVUM. It is FLAVUM flap. Thick fibers are seen in ligamentum flap. Okay. Now coming to the special stains. Special stains for the elastic fibers. One is the porcine with aldehyde fusion. Then you can see the Verhoff stain. Okay, the Verhoff stain. Okay, one thing I forgot to tell you is about the reticular fibers. Okay, they stain black with silver impregnation. Okay, silver impregnation. Okay, right? So this is about the fibers. Okay, now coming to the cells in connective tissue. Okay, there are two types of cell in connective tissue. Okay, one is the intrinsic cell. Okay, intrinsic cells, that means they will forms the intrinsic material. Okay, or the cells which forms the ground substance. Okay, cell which forms the intrinsic material, and this includes fibroblast, fibrocyte, and adipocyte. These are the cells which forms the intrinsic material. Then second one is the immune cells. Okay, like the lymphoblasts, lymphocyte. Okay, immune cells like macrophages and lymphocyte. So, right, I'll describe about this fibroblast. Okay, fibroblast. These are spindle shaped cells. Okay, spindle shaped cells which forms what? They forms the extracellular matrix. Okay, extracellular matrix of connective tissue. Okay, and they have a flat nucleus. Okay, spindle shaped cells with flat nucleus, right? Then coming to fibrocytes, these are nothing but inactive fibroblast. Inactive fibroblast. Okay, then adipocyte. Adipocyte, we know it is a fat cell. Okay, where the deposition of fat occurs, right? So since it is deposition of fat, which is a dye which we use, we can use benzene or xylene. Okay, what happens if you use benzene or xylene dye? The fat will get dissolved and you can see empty spaces. Okay, empty spaces. The adipocytes are present as empty space. There are two types of adipocytes. Okay, one is the yellow adipocyte and second is the brown adipocyte. The yellow adipocyte is seen in adults. Okay, adults, and brown adipocytes is seen in newborn. What is the difference? The yellow adipocytes, they are unilocular. Okay, unilocular. Okay, whereas the brown adipocytes, they are multilocular. Right? So brown adipocytes seen as multilocular. That is, the multiple fat droplets are seen. Okay, inside this cell. Okay, right? So this is about the cells. Okay, now coming to the classification of connective tissue. Classification of connective tissue. The connective tissues are divided into loose connective tissue and dense connective tissue. Loose connective tissue means the fibers are arranged in a loose manner. Okay, that includes areolar connective tissue, then adipose connective tissue, and reticular connective tissue. Okay, so here the connective tissues are arranged in a loose manner, whereas dense connective tissues are seen in dense regular connective tissue and dense irregular connective tissue. Okay, so we'll take one by one. First, the areolar connective tissue. Can you see here? So where can you see this areolar connective tissue? The areolar connective tissue is seen in the subepithelium. Okay, like the lamina propria. Okay, right, lamina propria. So that is where you can see areolar connective tissue. Okay, then reticular connective tissue. We have already said which type of fibers will be in reticular connective tissue. We will have reticular fibers, right? So, and you can see in the reticular connective tissue, see in the lymphoid organs except thymus. Okay, like lymph node, spleen. Okay, so here on you can see the lymphoid reticular connective tissue, right? Next is the dense connective tissue. This is the dense regular connective tissue. This is the dense irregular connective tissue, right? So where you can see dense, can you see here the fibers are arranged in a regular fashion, right? So dense regular connective tissue is present in the tendons, ligaments, and aponeurosis. Okay. Dense irregular connective tissue. Dense irregular, they are present in the dermis, okay, and in the periosteum or the organ of capsule, okay, where you can see dense irregular connective tissue. Understand? Right. Now, with this we have completed the connective tissues, right? Now we are moving on to the cartilages, okay, cartilages. So cartilages, what are the components of cartilage? Cartilage contains cells plus the matrix. Okay, we know there are cells, and the cells of the cartilage is called as what? They are called as chondrocytes. Okay, the cells are called as what? Chondrocytes. And the matrix will contain the ground substance, right? And the fibers. Okay, which type of fibers? The fibers can be collagen fibers and elastic fibers. Okay, elastic fibers. So this is the components of the cartilage, cells as well as matrix, right? There are 3 types of cartilage. Okay, 3 types of cartilages. One, you have the first one, hyaline cartilage. Then you have the elastic cartilage. Then you have the fibro cartilage. So we will take one by one. Hyaline, elastic, and fibro cartilage. So first, this is the hyaline cartilage. Can you see here hyaline? So tell me the arrangement. Here the chondrocytes are arranged in groups. Okay, they are arranged in groups. Second thing, what you need to know, look at the ground substance. The ground substance or the matrix, they are shiny and glossy. Okay, they are shiny and glossy. The ground substance are shiny and glossy, right? Then you can see the perichondrium. Okay, can you see the perichondrium will be present? Perichondrium will be present. And which is the type of collagen that is present in this hyaline cartilage? It is type 2 collagen. We have already discussed when we discussed about the types of collagen, right? So this type 2 collagen will be present in the hyaline cartilage, right? You know why this ground or shiny appearance in case of this cartilage? Because the refractive index of the ground substance, okay, or the matrix is equal to the refractive index of collagen. That is why you get a shiny appearance, okay. That's why you get a shiny appearance in case of hyaline cartilage. The refractive index of the ground substance is equal to the refractive index of the collagen, okay. This is about the hyaline cartilage. Now moving on to the elastic cartilage. Before that, I forgot to tell you the location where you can see hyaline cartilage. Hyaline cartilage is present in the respiratory cartilage, that is up to which level? Up to the bronchus. Up to the bronchus level you can see respiratory cartilage. Okay, so that is made up of which type? Hyaline cartilage. Then the embryonic cartilage. Then the epiphyseal cartilage. Okay, epiphyseal cartilage. Then the articular cartilage. Okay, they are what? They are hyaline cartilage. So the respiratory cartilage, embryonic cartilage, epiphyseal cartilage, and articular cartilage, they are hyaline cartilages. Understand? Second is the elastic cartilage. Okay, elastic cartilage, here also the chondrocytes are arranged in groups. Okay, chondrocytes arranged in groups. Then the elastic fibers, okay, elastic fibers are more than the collagen fibers. Okay. But what type of collagen fibers is present? It is type 2 collagen. Type 2 collagen. Right? So type 2 collagen is present. Then they will also have perichondrium. Okay. So the perichondrium is present in case of elastic cartilage. Now, where is the location? Where you can see elastic cartilage? Everything that comes with E. Okay. The ear pinna. Then the external auditory canal, the eustachian tube, okay, eustachian tube, then the epiglottis, okay, will have what type of cartilage? Will have the elastic cartilage, okay. Then the laryngeal cartilages, that is the tip of arytenoid, corniculate and cuneiform. Okay, so these are what type of cartilage? Elastic cartilage. Okay. Now moving on to the fibrous cartilage. So here, this can you see the cells are arranged in rows? Okay, cells are arranged in rows. And in between, can you see the collagen fibers arranged? So in between these rows, you can see the collagen fibers. So what type of collagen fibers will be seen? It is the type 1 collagen. Okay, type 1 collagen fibers are present. Okay, and the perichondrium is absent. There is no perichondrium in the fibrous cartilage. Okay, no perichondrium in the fibrous cartilage. Okay, so where is the location? If you take the locations, it is present in the articular disc. Okay, the articular disc, the intervertebral disc, okay, then the glenoid labrum, just at the shoulder joint, okay, then at the insertion of tendons, okay, insertion of tendons. If you have cartilages, then it will be fibrous cartilage, okay, then at the pubic symphysis, okay. So this is the location of the fibrous cartilage. Okay, so with this we have completed the cartilages. Okay, we have seen the classification, the fibers, the cells, everything. Okay, now we are moving on to the muscles. Okay, very simple topic. We know there are 3 types of muscle. This is the skeletal muscle, this is the cardiac muscle, and this is the smooth muscle. Okay, so from here it's like this, right? Is arranged in the form of fibers. Okay, the skeletal muscle is arranged in the form, and you can see nucleus. Okay, the nucleus will form what? The nucleus will form syncytium. That means can you see like a single cell with numerous nucleus? That is what we see. Okay, even though it is divided into different sarcomeres, right? Then syncytium is seen. Then can you tell me? It has dark and light bands. The skeletal muscle, okay, dark and light bands, okay, which is called as the A band and I band, anisotropic band and isotropic band, okay. Then which is forms an isotropic and isotropic, which we have already discussed in the physiology. Okay, the structure of muscles we have already discussed in the physiology, where you can see the H zone, Z line, what is H band, okay, that we have already completed there. Okay, now coming to the cardiac muscles. Cardiac muscles, they will form what? They will form branches, and these branches will anastomose each other. Then there is intercalated disc. Okay, intercalated disc, and in which type of nucleus? They have round nucleus. Okay, round nucleus. Then smooth muscles, they are spindle-shaped cells. Okay, spindle-shaped cells, and flat nucleus. Okay, with flat nucleus. Okay, location, skeletal muscles, these are the voluntary muscles. Cardiac muscle is seen in the heart, smooth muscles in the hollow visceral organs. Okay, so we know everything about the muscles. Okay, now about the fascia. Okay, we know the whole muscle is covered by which fascia? It is covered by epimysium. Okay, the whole muscle is covered by epimysium, right? The muscle fasciculus. Fasciculus is covered by perimysium. And the muscle fiber is covered by endomysium, which we have discussed. Okay, everything is discussed in physiology. Okay, right. So this is about the muscle. So this is about the general histology. Now we are moving on to each systems. Okay, first we are going to discuss about the nervous system. What forms the nervous system? We know there is the neuron. Okay, so the parts of neuron. One is the cell, body, or soma. Okay, this is the part of the neuron with cell membrane and cytoplasm. Okay, and this cell body have something called as cell granules. Okay, which will take up the basic dye. Okay, and appears as blue. Okay, so if it's large blue like granules, they are the cell granules. Then the cell body have large amount of endoplasmic reticulum. Okay, what does that mean? That means it is a size where increased protein synthesis occur. Okay, increased protein synthesis occur. Then you have neurofibrils, which are the thread-like structures that is seen in the cytoplasm. Okay, they are nothing but the microtubules and microfilaments. Then you have this cell process. Okay, cell process. There are two types of cell processes or the neurites. Okay, one is the dendrites and next is the axon. Okay, dendrites are short processes, whereas the axon is the long process. Dendrites is vary in thickness. Okay, they vary in thickness, whereas the axon is uniform in thickness. Okay, dendrites contains cell granules, whereas in case of axons, the cell granules are absent. There is no cell granules. Okay, then conduction is from here. The conduction from dendrite to cell body. Okay, here the conduction from cell body to axon. Okay, that is the direction of conduction. Okay, now there is an area from where this axon arises. Okay, the area from where the axon arises, that is called as the axon hillock. Okay, this area is cell granules are absent in this area. Right, now coming to the myelination. We know how myelination occurs, or which are the cells that helps in myelination. In CNS, the oligodendrocytes are responsible for myelination. Okay, whereas in peripheral nervous system, it is the Schwann cells. Okay, now how this myelination occurs? The axon will invaginate into the Schwann cell. If this is the Schwann cell, this axon will invaginate into the Schwann cell. Okay, and this axon will be suspended in the Schwann cell with the help of a mesoaxon. Okay, right, and this mesoaxon will extend and winds around the axon. Understand what is happening? The first one is axon invaginate into Schwann cell. Second, there is a suspension. Okay, the suspension of the axon is called as the mesoaxon, which will wind around the axon. Right, now over this winding, lipid deposition occurs. Okay, lipid deposition occurs over this winding, resulting in the formation of myelin sheath. Understand how myelination occurs? Right. This Schwann cells, okay, Schwann cells can only produce myelin sheath to a short distance. Okay, they produce myelin sheath for a short distance. That means there will be a gap between each myelin sheath. Okay, between each myelin sheath. And that gap is called as what? It is called as node of Ranvier. It is called as node of Ranvier. Okay. And the conduction occurs in case of a myelinated axon is what type of conduction? This is the node of Ranvier, and it is the saltatory. Okay. So this is about the structure. Now, next is the layers of cerebrum. If you take a cerebrum, these are the 6 layers of cerebrum. Before that, let me tell you the cells in the cerebrum. There are actually 2 types of cell. One is the pyramidal cell. Second is a stellate cell. Okay. The pyramidal cells, these are the large cell that will extend up to the whole thickness of the cerebrum. Okay. Now, these are triangular in shape. Okay. Triangular in shape, such that the dendrite will arise from the apex. Okay. This is the dendrite will arise from the apex, and the axon is formed from the base. Understand? This is a pyramidal cell. Stellate cell, they are star-shaped cell. Okay. And they are small cells. They are multipolar cells. Okay. Multipolar cells. Okay. So this is the 2 types of cell in motor cortex. That is, pyramidal cells are more than the stellate cell. And in sensory cortex, stellate cells are more than the pyramidal cells. Remember this. Okay. In motor cortex, it is pyramidal cells are more compared to stellate cell, and in sensory cortex, stellate cells are more compared to the pyramidal cells. Okay. Now coming to the layers, there are 6 layers. Okay. Cerebrum has 6 layers. Okay. Which are the layers? Can you see? The first layer is the molecular layer. Okay. Which contains the horizontal fibers. Okay. Molecular layer contains the horizontal nerve fibers. Okay. Nerve fibers. Right. Second layer is the external granular layer. Okay. External granular layer. Okay. Granular layer means what? The stellate cells are also called as the granular cells. Okay. So it will contain stellate cells. Okay. Majority stellate cells. Okay. Then you have external pyramidal layer. Okay. External pyramidal layer, where you can see small or medium sized pyramidal cells. Okay. Small or medium sized pyramidal cell. Then you have the internal granular layer. Okay. Internal granular layer, which contains the stellate cells. And you have internal pyramidal cells. Okay. Which has the large pyramidal cells. Okay. And the 6th layer is called as the inner layer or plexiform. Okay. Plexiform layer. Okay. Plexiform layer. That means it contains all types of cells. Okay. All types of cells. So I'll show you. This is the first layer, that is the molecular layer. Then you can see the granules. External granular layer. External pyramidal layer. This is internal granular layer. Internal pyramidal layer, and this is the plexiform layer. Okay. So this is the layer of cerebellum. Okay. With this, we have completed the nervous system. Okay. Next is the endocrine system. In the endocrine system, we will be discussing pituitary gland, thyroid, and adrenal gland. Okay. So first one, the pituitary. We know that the pituitary gland has 2 lobes. Okay. One is the anterior lobe, which is called as the adenohypophysis. and we have the posterior lobe, which is called as the neurohypophysis. Okay, so this adenohypophysis, this has two types of cells. Okay, two types of cells. I'll write in the next column. Okay, one is the chromophil cells and next is the chromophobe cells. Okay, this chromophil cells is divided into acidophil cells and basophil cells. Okay, it has acidophil cells and basophil cells. So the acidophil cells includes somatotrophs. It secretes what? This one. Somatotrophs secrete the growth hormone. Okay. Somatotrophs, which secretes the growth hormone, and mammotrophs, which secretes prolactin. The basophil cells includes the thyrotrophs, corticotrophs, and gonadotrophs. Okay, the chromophobe. What is the difference between chromophil cells and chromophobe cells? The chromophil cells, they have granules. Okay, granules. Okay, in acidophils they have eosinophilic granules, and in basophil cells they have basophilic granules. Okay, but the chromophobe cells, they don't have, the granules are absent. Okay, no granules absent. Understand? So this is the anterior pituitary. Now taking the posterior pituitary, what is there in the posterior pituitary? The posterior pituitary has the axons that is coming from the hypothalamus. Okay, the axons that coming from the hypothalamus reaches the posterior pituitary. From which part of the hypothalamus? From the supranuclear, supraoptic, not nuclear, supraoptic and paraventricular. Okay, so the axons that is coming from the supraoptic nucleus and the paraventricular nucleus reaches the posterior pituitary, and they secretes two hormones. Which are the hormones? That is the vasopressin and oxytocin. Okay, so this is about the posterior pituitary or the neurohypophysis. Okay, so they contains the axon. What type of axon? They are unmyelinated axons. Okay, unmyelinated axons. Okay, this is about the pituitary. Now we are moving on to the thyroid gland. Thyroid gland is located in the neck region, right? It has two lobes, right and left lobe, which is joined by isthmus, that we know. If you take the thyroid gland, it is covered by a capsule. Okay, covered by a capsule. From the capsule, septum forms. Okay, the extension of the capsule called as septum, that will divide the thyroid gland into lobules. Okay, so each lobule will have aggregates of follicles. Okay, so this is the structure of the thyroid gland. If you take the thyroid gland, there are two types of cells. Okay, two types of cells in thyroid gland. Which are the types of cells? One is the follicular cells. Okay, follicular cells, and second is the parafollicular cells. Okay, follicular cells and parafollicular cells. Okay, now coming to the follicular cells. Okay, which type of cells? The follicular cell type is determined by the amount of the hormone secreted. Okay, if the thyroid gland is working normally, then the follicular cells will be simple cuboidal. Okay, what happens if there is scanty secretion or very less secretion? Then it will be squamous epithelium. Okay, and when there is excess secretion, increased secretion or active gland, they will have columnar cells. Okay, follicle will have columnar cells. Okay, so the follicular cells means what? The cells that lines the follicle. Okay, so here the follicular cells you can see, and inside you can see the colloid, right? So these are the follicular cells. Okay. Second one is the parafollicular cells. Okay, parafollicular cells or C cells. Okay, this is called as the clear cell. Okay, they are polyhedral in shape. Okay, and they, where can you see them? They are seen between the follicles. Okay, seen between the follicles. And what is the secretion? They secrete thyrocalcitonin. Okay, they secrete thyrocalcitonin. This is about the parafollicular cells. Okay, now we are moving on to the parathyroid gland. Parathyroid gland are actually 4 in number. We know they are 4 in number. 2 superior, 2 inferior, right? And here these cells are arranged in cords, okay? And in between these cords you can see sinusoids, okay? In between these cords you can see sinusoids, okay? There are 2 types of cells. One is the chief cells and next is the oxyphil cells. Okay, so 2 types of cells, chief cells and oxyphil cells. Okay, chief cells is also called as the principal cell. Principal cell. Okay, principal cell, and they secrete the parath hormone. Okay, parathyroid hormone, right? Oxyphil cells actually they are less in number and their function is not known. Okay, function is unknown. So this is about the parathyroid gland. Now we are moving on to the adrenal gland. Adrenal gland, you know, adrenal gland, it has a capsule. Under the capsule, you have the zona glomerulosa. Then you have zona fasciculata, zona reticularis, and you have the adrenal medulla. So this is the arrangement of adrenal gland. Okay, so this much, that is the whole thickness of the adrenal gland. Okay, only 1/10 of the adrenal gland is by medulla. That means the thickness of cortex is 10 times that of medulla. Okay, thickness is cortex is to medulla is 10:1. Understand? The cortex is 10 times thicker than the medulla. Okay, now what are the secretions? We know zona glomerulosa, it will secrete mineralocorticoid, fasciculata, glucocorticoids, and reticularis, sex corticoids. And medulla, adrenal medulla will secrete what? They will secrete norepinephrine, right? So if you take the adrenal medulla, they have what type of cells? They have the post-ganglionic no, not postsynaptic, they have the post-ganglionic sympathetic fibers. Okay, they have post-ganglionic sympathetic fibers. Post-ganglionic sympathetic fibers. Okay, right term which we need to discuss here is the chromaffin reaction. What is meant by chromaffin reaction? If we fix the adrenal cells, okay, adrenal medullary cells, and if we add solution containing chromium salts, you can see yellow granules in cytoplasm. Okay, this is called as what? This is called as chromaffin reaction. Okay, now what are Apert cells? These are scattered neuroendocrine cells which shows chromaffin reaction. Chromaffin reaction. And what does they do? They will take up, these cells will take up amine precursors okay, and it will be converted to hormones or peptides by decarboxylation. Such types of cells are called as the Apud cells. An example, you have the adenohypophysis in pituitary. You have the neurohypophysis. You have the chief cells of parathyroid. Then C cells of thyroid. Then the adrenal medullary cells. Glomus cells in the carotid body. Melanocytes. Then the JGH, extraglomerular apparatus cells in the kidney which secretes rain. Okay, so these are the examples of Apud cells. Okay, I told you the properties of Apud cell. So this is about the adrenal gland, okay, or the endocrine system. Now we are moving on to the lymphatic system. So here you need to know about the lymph nodes, the tonsils, the spleen. So these are the structures which we are going to discuss mainly. Okay, first one, lymph nodes. One more thing, thymus. Okay, you need to study about thymus also. So lymph nodes, actually lymph nodes is the shape is a bean-shaped structure. Okay, it has one concave surface. So this is convex surface. One convex surface and one concave surface. Okay, convex surface and concave surface. Convex and this is concave surface. Okay, so the numerous afferents will enter into the lymph gland via this convex surface. Okay, numerous afferents will enter into the lymph nodes via this convex surface, and a single efferent will pass down via this concave surface. Understand? Now, if you take the lymph gland, it has a capsule. Okay, and the outer layer is the capsule. Under the capsule, you have the subcapsular sinus, which is filled with the lymph. Okay, then you have the cortex, paracortex, and medulla. Okay, so the lymphoid tissue, okay, the lymphoid tissue is arranged in the form of lymphoid follicles in the cortex. Okay, so whereas they are arranged in the form of cords in medulla. Okay, so the lymphoid tissues are arranged in the form of lymphoid follicles in the cortex and as arranged as cords in medulla. Right? This is about the lymph node. Now we are moving. Can you see here? This is the cortex, paracortex, then medulla. This is the germinal centers. Okay, germinal centers. This is the subcapsular space. This is the capsule, and this is the trabecule. Okay, right? Now moving on to the spleen. So, sorry, thymus. I'll start with thymus, then I'll go to spleen. So thymus is actually a primary lymphoid organ. Okay, it is a primary lymphoid organ. Okay, and it is responsible for the production of T lymphocytes. Okay, T lymphocytes, right? So if you take the thymus, it has a capsule. Okay, it has a capsule, and the lymphoid tissue are arranged in the form of lobules. Okay, lobules in thymus, right? So each lobule will have their own cortex and medulla. Okay, each lobule will have their own cortex and medulla. Okay, right? So in cortex, the lymphoid tissue or the lymphocytes are more. Okay, whereas there will have few reticuloepithelial cells. Okay, few reticuloepithelial cells. In medulla, it will reverse. Okay, there will be less lymphoid tissues and more reticuloepithelial cells. Okay, lymphocytes less and more reticuloepithelial cells. Okay, so this is about the thymus. I'll show you. Can you see here? This is the thymus, right? And the lymphoid tissues are arranged in the form of follicles. Okay, each follicle will have their own cortex and medulla. Okay, and can you see a trabeculum, which trabecular septum, which extend into the medulla, into the thymus from the capsule? That is also present. One thing which you need to know here is about the Hessel's corpuscle. Okay, Hessel corpuscles. Okay, Hessel's. These are actually the degenerated cells. Okay, degenerated cells which appears pink in color. Okay, pink in color. Their function is unknown. Okay, still unknown what they do. Okay, right. So this is about the thymus. Now we are going on to the spleen. Spleen is a secondary lymphoid organ. Okay, it has a capsule. Okay, from the capsule, it has a capsule. Okay, from the capsule, the trabeculum will arise. Okay, trabeculum and the splenic arteries. Okay, the splenic arteries will enters into the trabeculum. Okay, splenic artery will enters into the trabeculum, forming trabecular arteries. Okay, this trabecular arteries later forms the arterioles, and they will give rise to sinusoids. Okay, this is the this is how the division occurs. Right now, if you take the spleen, can you see here? This is the spleen, right? So spleen has a white pulp and a red pulp. Okay, these areas are the white pulps. Okay, can you see here? This is the white pulp. Okay, and this is the red pulp. Okay, now let's white pulp. They are actually only 10 to 30%. Okay, they are only take up around 10 to 30% of the spleen, and they has increased number of WBCs. Okay, they has, can you see here, more WBCs. Okay, they have cortex and medulla. Okay, the cortex is darkly stained. Okay, it is darkly stained, and medulla is pale. Okay, and why darkly stained? Because it has B lymphocytes. Sorry, it has T lymphocytes. Sorry, the cortex has T lymphocytes and the medulla has B lymphocytes. Okay, cortex has T lymphocytes and medulla has B lymphocyte. And there is a eccentrically spaced central arteriole. Okay, eccentrically spaced placed central arteriole. This arteriole is surrounded by a periarteriolar lymphatic sheath, which contains what? Which contains the T lymphocytes. Understand? I'll show you. Can you see here? This is the darkly stained periphery and this is the lightly stained medulla. Okay, and this is the eccentrically placed arteriole, right? And that eccentrically placed arteriole has a periarteriolar lymphatic sheath. Okay, this is about the white pulp. The red pulp, it has more number of RBCs. Okay, and it is around 70 to 90% of the spleen is this red pulp. Okay, it has what? It has sinusoids. Okay, sinusoids. Okay, so this is about the red pulp and white pulp. Okay, right. Now we are moving on to the next lymphoid organ, that is tonsil. How many tonsils are there? We have lingual tonsil, you have pharyngeal tonsil, and there is palatine tonsil. Okay, I'll just speak something about the palatine tonsil. Okay, this palatine tonsil, can you see the medial side of the tonsil have crypts? Okay, they have crypts. Okay, so this crypts, and under this crypts the lymphoid tissue is arranged in the form of follicles. Okay, and the crypts, the epithelium of these crypts is non-keratinized squamous epithelium, stratified squamous epithelium, non-keratinized stratified squamous epithelium. This we have already seen, right? So this is about the tonsil. Now finally coming to the last one, that is malt. Malt is a mucosa associated lymphoid tissue, and this malt usually seen in GIT, right? Which layer? In the lamina propria. Sometimes this malt is present where? This malt can extend into submucosa, but rarely, okay, rarely it can extend into submucosa, where you can see in the vermiform appendix, okay, where this malt will extend into the submucosa. Okay, so this is about the lymphoid tissue. Okay, coming to the GI tract. Okay, so there are 4 layers in the GI tube or the gut tube. Okay, these layers are mucosa, submucosa, muscularis externa, and serosa. Okay, so these are the layers of the gut tube. Okay, right? So if we take one by one, if we take the mucosa is again divided into 3. Okay, the first part is the epithelial part, mucosal epithelium. Then second, you have the lamina propria, and third, you have the muscularis interna or muscularis mucosa. Okay, muscularis interna or muscularis mucosa, right? So these are the 3 parts of the mucosa. Now coming to the submucosa. Submucosa is the strongest layer. Okay, it is the strongest layer in GIT. Okay, and a nerve plexus runs along the submucosa, and that is called as the Meissner's plexus. Okay, Meissner's plexus. Esophageal glands and the duodenal glands, okay, which is called as the Brenner's gland, is seen in the submucosa. So esophageal glands and duodenal glands are present in the submucosa. Next is the muscularis externa. There are 2 layers of muscles. Okay, you have an inner circular muscles and an outer longitudinal muscle. Okay, outer longitudinal muscle. What you need to know here is that in between these muscles, okay, between these muscles, a nerve plexus runs, that is called as the Orbach's plexus. Okay, Orbach's plexus runs between the inner circular and outer longitudinal muscle. Okay, and the last layer is the serosa. The serosa layer, it is present where peritoneum is there. Okay, the part of the GI tube which is covered by the peritoneum, they have the serosa. Can you tell me a part which is not covered by the peritoneum? It is esophagus. Okay, esophagus. So what is, they don't have no serosa. Okay, they don't have serosa. Instead, what we'll call the outer layer is called as the adventitia. Adventitia. Okay, if there is no serosa, then the outer layer will be adventitia. Understand? So this is the layers of GI tube. Now we'll take each part. The first part we will discuss is the esophagus. The mucosa of the esophagus, which is the mucosa, the mucosal epithelium. Mucosal epithelium is stratified, non-keratinized squamous epithelium. Okay, stratified, non-keratinized squamous epithelium. At the end of the esophagus, where the esophagus meets the stomach, there is a transitional zone. This is called as what? This is called as the gastroesophageal junction. Okay, gastroesophageal junction, okay, where the transition of the epithelium occurs, okay, till this line, this transitional zone, it is stratified squamous epithelium. Below that there will be columnar epithelium, okay, columnar or cuboidal epithelium, and this is the place where the epithelium transforms, okay. And I told you this transitional zone is subjected to what? It is subjected to metaplasia. And can you tell me which is the most common metaplasia at the gastroesophageal junction? It is the intestinal metaplasia, okay, intestinal metaplasia. That is at the junction, the squamous epithelium is converted into columnar epithelium with goblet cells, okay, with goblet cells, okay, that is called as the intestinal metaplasia. And this intestinal metaplasia will lead to adenocarcinoma, increased risk of adenocarcinoma, and this condition is called as what? It is condition is called as Barrett's esophagus, okay, this is called as Barrett's esophagus. Understand? This is about the esophagus. Now we are moving on to the stomach. Draw the stomach first, okay. This is the esophagus, okay. From esophagus you have the stomach. So this is the shape of stomach, right? And the part of the stomach that attached to the esophagus is called as the cardia, right? The above is the fundus, then you have the body, and last is the pylorus, okay. So this is the 4 parts of stomach, okay. And what can you see this glands of the stomach? The stomach or the gastric glands, they are present at the level of lamina propria, okay, mucosal layer, okay, lamina propria, right? Now I'll give the cells that is present in the gastric glands, okay. We'll discuss the cells that is present in the gastric glands, okay. First cells, it is the surface mucus cell, okay. These are the surface mucus cell or the first cell, okay. What they will secrete? They will secrete alkaline mucus, okay. They will secrete alkaline mucus, okay. Second cell are the mucus neck cells, okay. These are the mucus neck cells, okay, and they will secrete acidic mucus, okay. The third ones are the parietal cells, okay. The parietal cells, oh, they will secrete what? They will secrete HCl and intrinsic factor, okay. They will secrete HCl and intrinsic factor, and these cells will appear pink in color, and they have a fried egg appearance, okay, fried egg appearance. The fourth cells are the chief cells, okay. They are the chief cells or the oxyphil cells, okay, oxyphil cells, okay, and they will secrete pepsin and gastric lipase, okay. They will secrete pepsin and gastric lipase. They will take up their basophilic, okay, so they will appear as blue in color, okay. The chief cells will appear blue in color. And the fifth cells are the endocrine cells or the argentophil cells, okay, argentophil cells, and they will secrete what? They will secrete gastrin, okay, and this gastrin will helps in the secretion or will determine the secretion of the pepsin or the gastric juices, okay. And in the basal layer you have the stem cells, which is responsible for the mitotic activity, okay. So this is the cells in the gastric glands, okay. So you can see that the upper half has what? They has the mucus cells as well as the parietal cells, and the lower half has chief cells and the endocrine cells, okay. And remember the thing, there is no goblet cells in the stomach, no goblet cells, okay. So this is about the stomach. Next we are moving on to the intestine. So intestine has 3 parts, right? We all know the 3 parts of intestine. The proximal part is called as the duodenum, the jejunum, and ileum, okay. So what's the major function of this intestine? The jejunum and ileum are for absorption, okay. So they are designed for absorption, right? So the parts or the layers, 4 layers of GI tube is present in the intestine, right? So we'll see what is the difference between duodenum, jejunum, and ileum, okay. So this is the duodenum, okay. This is the mucosa, this is the lamina propria, Then this is the intestinal gland. Okay. Then you have the muscularis mucosa, and this is the submucous gland or the Brunner's gland. Okay, so what's the difference? The duodenum has Brunner's gland or the submucosal glands. Okay, this is the muscularis externa, the circular muscles. Okay, and this is the longitudinal muscles, and this is the serosa. So these are the layers of duodenum, and what is the specialty? It has Brunner's gland or the submucous gland. Right now, coming, this is the jejunum. Okay, jejunum, you can see finger-like villi. Okay, large finger-like villi. Villi as well as this is microvilli. So you can see the microvilli as well as the villi, large villi. That means major amount of absorption. Okay, major amount of absorption occurs at the jejunum. Okay, jejunum. So it is mainly specialized for absorption. Okay, and this is the I'll show. This is the mucosa. This is the lamina propria. This is the muscularis mucosa. Okay, this is the submucosa, and this is the muscularis externa, and last is the serosa. Okay, right? So you have to say that the prominent villi and microvilli in the jejunum will helps in the absorption of substances. Right? This is the ileum. So here also you can see these are the crypts. Okay, intestinal crypts. And what is the name of that intestinal crypts? It is called as the crypts of Lieberkühn. Okay, so in this crypts of Lieberkühn, we have the intestinal glands. Okay, these are the intestinal glands. Can you see here the intestinal glands? Okay, and this is the lamina propria. What you can see in the lamina propria? You can see the lymphocyte aggregates, and that is called as the Peyer's patches. Okay, the Peyer's patches. Okay, right. So this is present in the lamina propria, and this is the submucosa, and this muscularis externa and serosa. Okay, right. So you how we will identify each part? Duodenum has Brunner's gland, the prominent villi and microvilli in the jejunum, and the Peyer's patches in the ileum. Right. Now we'll see about the cells in the intestinal glands. Okay, cells in the intestine. So the prominent cell is the enterocytes. Okay, enterocyte. And what's the function of enterocyte? This cell helps in absorption. Okay. Then you have the goblet cells. Okay, you have columnar epithelium with goblet cells, and this goblet cells will do mucus secretion. Then you have Paneth cells. These are actually have role in immunity. Okay, they will secrete cytokines. Okay, cytokines. Okay, then you have the M cells or the microfold cells. They are nothing but the antigen presenting cells of the intestine. Then you have the neuroendocrine cells, which will secrete the hormones, and you have the stem cells, which has the mitotic activity. Okay, so this is about the cells of the intestine, right? If you take the duodenum, I told you there is Brunner's gland. Okay, Brunner's gland. What is the secretion of the Brunner's gland? The Brunner's gland will secrete urogastron or human epidermal growth factor. What is the function of this urogastron or HEGF? It will decrease the parietal cell activity. Okay, parietal cell activity. That means what? It will decrease the HCl production. Okay, then it will increase the mitotic activity. Okay, mitotic activity. That means it will helps in healing. Okay, that means it is actually a protective role in case of duodenal ulcer. Okay, duodenal ulcer, they have a protective role, which is the urogastron or the secretion of the Brunner's gland. Okay, so this is about the small intestine. Now we are moving on to the large intestine. Cells in the large intestine. There are mainly two types of cells in the large intestine. One is the colonocytes. Okay, colonocytes, what is the function of colonocytes? They will absorb the water, electrolytes, and vitamins. Okay, so this is the function of electrolytes. Second is the goblet cells. Okay, They will secrete mucus, and what does this mucus will do? They will lubricate, okay, the rectum, the anus, okay. So the lubrication is the main function of the goblet cells. So what you can say that the large number of goblet cells, can you see here the white cells? Okay, large number of goblet cells are seen in the large intestinal epithelium. Okay, right. Now I want to discuss something about the anal canal. You know, anal canal has a transitional zone. Okay, so the upper anal canal, okay, the upper anal canal is derived from the endoderm, right? So it will have the columnar epithelium. The lower anal canal, it is derived from the ectoderm. So it will have stratified squamous epithelium, right? So there is a line which separates the upper anal canal and the lower anal canal, that is the dentate line or the pectinate line. Okay, so this zone where you can see the columnar epithelium is the colorectal zone. Okay, right? And the region that is below or the region that is between this colorectal zone and the pectinate line is called as the anal transitional zone. Okay, that is the anal transitional zone. What is the feature? Here the cells can be cuboidal or columnar or squamous. Okay, the transitional zonal cells can be cuboidal, columnar or squamous. That is, it is a transitional epithelium, right? And below the pectinate line, you have this squamous epithelium. Okay, there is a one more line which will separate the lower distal part of the anus into two halves. Okay, one half you will have stratified squamous epithelium, and below that line you will have stratified squamous keratinized epithelium. Okay, one is non-keratinized and other is keratinized. Okay, and the line that separate the stratified squamous non-keratinized from the keratinized epithelium is called as the white line of Hilton. Okay, it is called as the white line of Hilton or the mucocutaneous line. Okay, so this transitional epithelium we have discussed, there is a high chance of metaplasia. Okay, so you need to know this difference. Okay, first you have the colorectal zone where you have columnar epithelium, then you have the transitional zone where you can have columnar, cuboidal or squamous epithelium, then you have the pectinate line or the dentate line, below you have the stratified squamous non-keratinized epithelium, then you have the white line of Hilton, and below the white line of Hilton you have stratified squamous keratinized epithelium. Okay, so this is about the intestine or large intestine. Now we are moving on to the liver. Moving on to liver, we know the liver is in the form of hepatic lobules, right? Hepatic lobules. What is the structure of hepatic lobules? It is one lobule will be in hexagonal shape, and you have the hepatocytes arranged in the form of cords, right? And in between these hepatocytes, what you have? You have sinusoids. Okay, you have sinusoids, and centrally you have a central vein. Okay, and at the peripheries of this hepatic lobules, you have the portal triad. The portal triad, you have the portal duct, portal vein, the bile duct, and the hepatic artery. Okay, so this is the content of the portal triad. Okay, so this is the structure of the hepatic lobules. Right now, if you take the blood supply, the liver or the hepatic lobules, they have dual blood supply. One pair of arteries or one pair of blood vessels, that is the hepatic artery, they will carry the oxygen. Okay, they will carry the oxygen and they will distribute the oxygen into the sinusoids. From this sinusoids, this blood will drain into where? Into the central vein. Okay, to the central vein. Then from the central vein to the hepatic vein to the sinusoid to central vein to hepatic vein to the inferior vena cava. Right. So this is the one blood supply. The second blood supply, it is via the portal veins. Okay, it carries nutrients. Okay, so the nutrients from the intestine via the enteroportal circulation. Okay, enteroportal circulation reaches the portal vein. from the portal vein. Can you see here? This is the portal vein. From the portal vein, it reaches the sinusoids. Okay, sinusoids or hepatocytes. Okay, then from there it is passed down to the central vein. Okay, then to the hepatic vein, to the inferior vena cava. Right. This is the two dual blood supply. So what is circulating? Blood is circulating in one from the hepatic artery. Can you see here the red one? And one from the portal vein. Then what is this green tube? That is the bile duct, right? So the hepatocytes will synthesize what? The hepatocytes will synthesize the bile. Okay, and that bile will be flowing in the opposite direction. Okay, it will be collected in the bile duct. Okay, it will be collected in the bile duct. Understand? This is what is happening right now. If you take the layers of the liver, okay, how are these cells in the liver is arranged? I'll show you. Can you see here? This is the cells that is arranged. Okay, so this is the hepatocytes. In between you have the sinusoids. Okay, what are the cells that is present in the sinusoids? You have the endothelial cells, then you have the Kupffer cells, and you have the stellate cells. Okay, so these are the three cells that is present in the liver. Okay, so hepatocyte, I told you their function is to produce bile. Okay, then the Kupffer cells, these are nothing but the macrophages. Okay, Kupffer cells are macrophages. Okay, and that the stellate cells, these are star shaped cells. Okay, some stellate cells will store vitamin A in them, and such stellate cells are called as the Ito cells. Understand? Ito cells. So these are the layers of cells in the liver, or the cells that is present in the liver. Okay, so mainly three types of cells: hepatocytes, Kupffer cells, and the stellate cell. Okay, right? And there is a space. Okay, this is the the hepatocyte epithelial space. Okay, and this space that is between and that space between the hepatocytes and the sinusoid, okay, or the capillaries, and that space is called as what? Is called as the space of Dissay. Okay, space of Dissay, right? And the space in the hepatic lobule. Okay, if you take the hepatic lobule, if this is the hepatic lobule, the space where you can see the portal triad, that is called as the space of Mann. Okay, space of. Okay, so these are the spaces and cells in the liver. Okay, now I'm moving on to the architecture, liver architecture. Okay, one architecture we have already studied, that is the hepatic lobule, right, which is hexagonal in shape, right? That is also called as the classic lobule. Okay, classic lobule, and I have already described. You have the codes of hepatocytes. Okay, center you have the central vein, peripheral you have the portal triad. Okay, this is the classic lobule. The second one is, you need to know, is about the portal lobule. Okay, portal lobule. This portal lobule is formed, or that structure is designed based on the bile drainage. Okay, the drainage of the bile. Okay, right? So how is it defined? Can you see here? So it is triangular in shape, right? And at the corners you have the central vein, and at the center you have the portal triad, right? So the liver, the bile drainage will be in this direction. Okay, so this is the portal lobule. Okay, portal lobule. Okay, which is triangular in shape, and at the center you have the portal triad, and at the sides you have the central vein. Okay, at the corners, side corners, you have the central vein. This is the central vein. Okay. Now the third form is this one. That is the liver acini. Can you see here? This is the liver acini. Okay, this is the third structure. Liver acinus. In liver acinus, it is based upon the blood flow. Okay, this division or this architecture is based upon the blood flow in the hepatic arteries and the portal veins. Okay, so if we take, this is one central artery, this is one portal triad, another central artery, another portal triad. So it will connect between the portal triad and the central arteries, right? So one side you will have this central, so not central vein. Okay, you will have this central vein, and the other side you will have the portal triad. Okay, right? And this area, this liver acinus is divided into 3 different zones. This is zone 1, zone 2 and zone 3. Zone 1, zone 2 and zone 3. Okay, understand? 1, 2 and 3. Okay, and show you. Can you see here? It is this purple area is zone 1, green area is zone 2 and yellow area is zone 3. Okay, and this division of zones is based on the blood circulation. Okay, so the zone 1 is highly vascular. Okay, zone 2 is moderately vascular and zone 3 is least vascular. Okay, so can you tell me where will be the ischemic injury more? The ischemic injury will be more in the zone 3. Okay, zone 3 increased ischemic injury. Okay, what about the toxic injuries? The toxic injuries will be more in the zone 1. Okay, toxic injuries will be more in the zone 1. Okay, so this is about the liver architectures. Okay, so 3 architectures we have discussed. Okay, now we will move on to the next system. So with this we have completed the gastrointestinal system. Now we'll be move on to the respiratory system. Respiratory system, I can say it starts from the larynx, right? So which is the flap that guards the trachea? That is the epiglottis, right? So this epiglottis has 2 surfaces. One is the oral surface and second is the laryngeal surface. Okay, this oral surface is lined by stratified squamous epithelium and the laryngeal surface is lined by the pseudo-stratified ciliated columnar epithelium, which is also called as the respiratory epithelium. Pseudo-stratified ciliated columnar epithelium is also called as the respiratory epithelium. Okay, understand? That lines the laryngeal surface of the epiglottis, right? So if you take the respiratory system, it has conducting part and respiratory part. So what are the structures that is included in the conducting part? The conducting part includes the trachea, the bronchus, the bronchioles, that is the up to this, that is the terminal bronchioles. Okay, that is the blue shade gives you the conducting part. Okay, up to the terminal bronchioles you have the conducting respiratory tract. Okay, and below the terminal bronchioles, that is the purple shade, that is the respiratory part. Okay, so the respiratory part includes the respiratory bronchiole, the alveolar duct, alveolar sac and the alveoli. Okay, so the conducting part includes the trachea, the bronchus, the bronchioles. Okay, up to the terminal bronchiole. Okay, so these are the cells that is present in the respiratory tract, right? If you take the layers of the respiratory tract, it has mucosa, mucosal layer, it has submucosal layer. Okay, mucous, then it has the cartilage. It is a cartilage that is present in the respiratory tract. It is the hyaline cartilage. Okay, hyaline cartilage, and this cartilage will be present up to bronchus, up to the level of bronchus. Okay, can you see here? Cartilage is present up to the level of bronchus. Okay. Similarly, the goblet cells, where can you see the goblet cells? The goblet cells are seen in the mucosa. Okay, they are also present up to the level of goblet cells, which is seen in the mucosa. They are present up to level of bronchus. Okay, the submucous layer. Okay, the submucosal glands. Okay, submucosal glands are also present up to the level of bronchus. Understand? Right now, if we take the epithelium, what is specialty? Up to bronchus, you have respiratory epithelium. This is a respiratory epithelium. It is the pseudo-stratified ciliated columnar epithelium. Pseudo-stratified ciliated columnar epithelium is present up to bronchus. Okay, the bronchioles. Sorry, bronchioles. The bronchioles, they have ciliated columnar epithelium. Okay, ciliated columnar epithelium, not pseudostratified. Okay, ciliated simple columnar epithelium is present in case of bronchioles. Right. If you come to the terminal bronchiole has simple cuboidal epithelium. Okay, simple cuboidal epithelium. Understand? What are the cells that is present in the mucosa of respiratory epithelium? Cells in respiratory mucosa. Can you tell me what are the cells? The cells include, first one, the goblet cells. We know what's the function of goblet cells. It is mucus secretion. Second one is the Clara cells. What are Clara cells? Okay, this Clara cells are actually present at the level of terminal bronchioles. Okay, from the terminal bronchioles. Okay, and they are associated with the detoxification of air. Okay, they will secrete some small amount of surfactant. Okay, and they can also act as the mitotic activity. They have some mitotic activity. Okay, this is about the Clara cell. Now you have the brush cells. Okay, their function is not known. Okay, or still unknown. Brush cells. Then you have the basal cells. Okay, basal cells, these are nothing but the mitotic cells. Okay, which will help in the proliferation of wear and tear. Okay, then you have the Pulchinski cells. Okay, these are nothing but the neuroendocrine cells. Okay, so these are the cells that is present in the respiratory mucosa. Okay, up to the bronchioles, or okay, up to the level of terminal bronchioles. Right now, if you take the alveoli is, and the structure is entirely different because we know it is stratified squamous epithelium. Now, what are the cells that is present in the alveolar mucosa? This include type 1 nematocyte. Type 1 nematocyte, what is the function? Gaseous exchange, right? Gaseous exchange. Then you have type 2. This type 1 nematocyte is 95%. Okay, 95% are type 1 nematocyte. Okay, then you have type 2 nematocyte, and we know type 2 nematocyte is associated with surfactant production. Okay, then you have alveolar macrophages. And you have alveolar pores of form. Okay, so these are the structures that you can see in the alveolar mucosa, right? So if you ask me what are the difference that occurs when, or what are the structures that is present till bronchi? Structures that is present till bronchi, I can say the hyaline cartilage that is present till bronchi, the respiratory epithelium that is present till bronchi, then the submucosal glands that is present till bronchi. Now, what happens to these substructures? After or below the level of bronchi, the hyaline cartilage will disappear and they'll be replaced by smooth muscles. Okay, so if you take the bronchioles, okay, they will have smooth muscles instead of cartilage. The respiratory epithelium will be transformed as ciliated columnar initially. Then they will be forming simple columnar, and by reaching the terminal bronchiole, they will form simple cuboidal. Okay, so the terminal bronchiole have simple cuboidal epithelium. Understand? And so this is about the respiratory system. Okay, now we moving on to genitourinary system. Okay, genitourinary system. So first I'll discuss about the renal tubules. Okay, renal tubules. What about the, we know Bowman's capsule, it is lined by what type of epithelium? Simple squamous, because it is for exchange. Okay, then you have the PCT, proximal convoluted tubule, it is lined by ciliated cuboidal epithelium. Okay, ciliated cuboidal epithelium, right? Then you have the thick descending limb. Okay, thick descending limb is also lined by the ciliated cuboidal epithelium. Okay, thick descending limb of loop of Henle, it is lined by ciliated cuboidal epithelium. The thin part of loop of Henle, okay, the thin part of loop of Henle, this is lined by squamous epithelium. Why? Because exchange of substances with the vasa recta. Okay, that is why thin part of this loop of Henle is lined by squamous epithelium. Then the thick ascending limb, it is lined by cuboidal epithelium. Okay, and DCT also lined by cuboidal epithelium. Okay, if you take the collecting duct, the tip of collecting duct, from the tip of collecting duct, what starts? The transitional epithelium starts. Okay, transitional epithelium starts from the tip of the collecting duct, and this transitional or the urothelium, it will extend up to the urethra. Okay, so you have the collecting part of the kidney, the whole collecting part, the renal calyces, renal pelvis. Okay, I have explained when I explained the transitional epithelium. The renal calyces, renal pelvis, the pyramids, the ureter, urinary bladder up to the urethra, you have transitional epithelium. Okay, so in case of males, now we are moving on to males. In case of males, the up to prostatic urethra, the prostatic urethra is, it is urothelium. Okay, but the membranous and the penile urethra, both are stratified columnar epithelium. Okay, this is about the male. Now moving on to the females, their fallopian tube is lined by ciliated columnar epithelium. Okay, because the cilia will help in the movement of, right? The uterus, the part of the uterus that is associated with the fallopian tube, they will be lined by ciliated columnar epithelium, and the other part which is lined by simple columnar epithelium. Okay, simple columnar epithelium, right? Moving on to the cervix, the upper cervix is lined by the columnar epithelium, simple columnar epithelium, and the lower cervix, it is lined by stratified squamous non-keratinized epithelium. Okay, so here also you have one, which zone? The transitional zone. Okay, at the junction of the upper and lower cervix, you have the transitional zone, and this transitional zone is the place of metaplasia, increased chance of metaplasia, and will lead to what? And will lead to cervical cancer. Okay, cervical cancer. Okay, so this is about the cervix. Then you have the vagina, which is lined by stratified squamous non-keratinized epithelium. Okay, so this is about the female system, male system, and the kidney. So with this, we have completed the histology part. Okay, so thank you everyone.